Unisexual means either the male reproductive organ, that is the anther, will be present, or the female reproductive organ, the gynoecium, will be present. The pink flower, one of the F1 plant is self with another F1 plant, that is pink flower plant self with a pink flower plant itself. Mice infected with the R strain do not develop pneumonia because R strain is a avirulent strain, and R strain infected into the mice will live, they do not die. Hello everyone, I am Dr. Vidhyaashram, Biology Faculty at Vidhyaashram Pre-University College, Temple of Excellence, Mysuru. So in this session, which is a continuation of the previous two sessions wherein we had discussed the model question paper consisting of 15 MCQs, 5 fill in the blanks, 2 marks questions and 3 marks questions. So we will continue with discussing about part D, which consists of 5 marks question in this particular session. So in the previous session, we had learned 2 mark questions and we had learned 3 mark questions. So in this session, we'll start with 5 mark question, that is the last part in the question paper. So answer any 4 of the following questions in about 200 to 250 words wherever applicable. So the first question, explain the different outbreeding devices developed by flowering plants. So this is also one of the most common questions that keeps appearing in the question paper. Outbreeding devices. So outbreeding devices are usually developed in order to prevent self-pollination and to promote cross-pollination, wherein plant itself, they naturally develop these outbreeding devices. So what are those? We will look into. So the devices to avoid self-pollination and to encourage cross-pollination are called as outbreeding devices. First one is pollen release and stigma receptivity are not synchronized. That is, if the pollen grains mature, the stigma will not be mature enough to receive the pollen grain. Or sometimes the stigma might become mature enough to receive the pollen grain, but the anther would have not released the pollen grain yet. So that is one of the mechanism. So next, outbreeding devices. In some other species, the anther and stigma are placed at different position so that the pollen cannot come in contact with the stigma. Sometimes what happens, if the stigma is of a very high position and the anthers are in a slightly lower position, then it is difficult for pollen grains to go from the anther and fall onto the stigma, which is slightly above the anther. So that is also one of the outbreeding device mechanism. Next, the third device to prevent inbreeding is self-incompatibility. This is a genetic mechanism wherein self-pollination will not happen even though the pollen grain fall on the stigma. The stigma will not allow the pollen grain to germinate. So this is also one of the outbreeding devices. Next one, in order to prevent self-pollination, unisexual flowers will be produced by the plant. So we all know that flowers are of bisexual and unisexual types. Bisexual means the male and the female reproductive organ will be present in the same flower. Unisexual means either the male reproductive organ, that is the anther, will be present, or the female reproductive organ, the gynoecium, will be present. Now here what happens, because it is a unisexual plant, only the male plant and the female plant will be there. So obviously cross-pollination needs to happen. So that is one of the outbreeding devices. The next one is, in several species such as papaya, male and female flowers are present in different plants. That is, each plant either male or female. That is dioism. This condition prevents autogamy and geitonogamy as well. So these are different methods in which plants promote cross-pollination and prevent self-pollination. Because it's for 5 marks, 5 outbreeding devices need to be mentioned here. Moving to the next question. Draw a neat label diagram of the sectional weave of mammary gland. It's a 5 mark question. So therefore, a total of 10 labeling should be put here. So this is the NCERT diagram of the mammary gland. So you have to draw exactly and label it without any spelling mistakes. Moving further. Schematically represent incomplete dominance by taking inheritance of flower color in Snapdragon as an example. So what do we mean by incomplete dominance? Incomplete dominance is a condition wherein both the parents will get expressed in the progeny. That is, neither of the parent is dominating one another. The dominant gene is not capable of completely dominating the recessive gene. So the characters mix up and ultimately we get pink color flowers. So that is about incomplete dominance. So first we'll write the definition. Incomplete dominance is a condition that sometimes the F1 shows phenotype that does not resemble either of the two parents and it has a in-between characteristic. So example, inheritance of flower color in dog flower, which is also called as snapdragon or anthurium species. So in a cross between true breeding red flower. So here we need to take the parents which are true breeding red flowers, which is represented by capital RR, and true breeding white flowers, which is represented by small rr. So the F1 will be capital R and small r, and either according to the test cross, or if we are doing any cross, that is Mendel's monohybrid experiment and all that, according to that we should have got a red colored flower, right? Because it is red is dominating here, but because it is incompletely dominant, we are getting a mixture of characteristic wherein the F1 progeny will have a pink color flower. So when the F1 was self-pollinated, the F2 resulted in the following ratio, that is 1:2:1, wherein one red flower, two pink flowers and one white flower will be seen in the F2 generation. So in the F2 generation, the phenotypic ratio changed from 3:1, that is dominant is to recessive ratio. Here factor R was not completely dominating over the small r, that is red was not completely dominating over the white flowers. Therefore, we could get an intermediate character which is incomplete dominance, that is a pink color flowers. So this is the schematic representation. So explanation needs to be done and schematic representation has to be put. So first they'll take the genotype of the parents, wherein they have taken the red flower, the white flower. Red is represented by dominant capital RR, white is represented by recessive small rr. Here the gametes are capital R and small r, and F1 generation they'll have a combination of capital R and small r, which is pink flowers. Next, selfing is done, that is the pink flower, one of the F1 plant is selfed with the another F1 plant, that is pink flower plant is selfed with a pink flower plant itself. So therefore, the resulting F2 generation will have a ratio of 1:2:1, that is red, 2 pinks and 1 white. So this is the schematic representation. So moving further, the next experiment, explain Griffith's transforming principle experiment. So transforming principle experiment to show how the R strain turned, which is A virulent, it turned into a S strain and it could cause infection in the mice, is what the experiment talks about. So here the answer is Streptococcus pneumonia bacteria are of two types. One is called as the S strain, which is virulent in nature, and the other one is the R strain, which is avirulent or non-virulent in nature. S strain, it is called as smooth strain. Why? Because it has a smooth, shiny appearance because of the presence of a mucus coating which consists of a polysaccharide, whereas R strain do not have this polysaccharide or the mucus coating, and therefore it is called as the rough strain. After writing that, we have to write about when each of these strain was injected into the mice, what happened to the mice. So mice which was infected with S strain died from pneumonia infection. That is, when we inject S strain into the mice dies. Next, mice infected with the R strain do not develop pneumonia because R strain is a avirulent strain, and R strain infected into the mice will live, they do not die. Now next what Griffith did was, he infected the mice with heat killed S strain. Now when we say it is heat killed, this will become avirulent or it will become non-virulent. And the mice did not develop any pneumonia because the S strain, we have heat killed it, we have made it avirulent. So next what he did was, he took a mixture of heat killed S strain and live R bacteria, that is Streptococcus pneumonia bacteria, and injected into the mice. The mice was supposed to live. Why? Because S strain heat killed means it is avirulent, which is not capable of causing infection, and R strain also is avirulent, not capable of causing infection. Then how did the mice die? But astonishingly, the mice had died. How did it die? So just writing this is not enough. You have to give a conclusion, like how did the mice die? Why did it die? Even though the A virulent, S strain and the R strain were injected into the mice. So Griffith concluded that some transforming principle transferred from the S strain to that of the R strain and made the R strain synthesize the polysaccharide mucus coat, making it virulent, and that is why the mice died. So that is how you need to conclude it. If you write so much, you will get 5 marks. So next question, what are the functions of? So they have given here 5 genes, that is I gene, LAC-Z gene, LAC-Y gene, LAC-A gene and inducer. Can you tell me like exactly what is this? This is regarding in the presence of an inducer, lactose inducer, and in the absence of an inducer, that is lac-operon concept. So it is that particular experiment. So in that experiment, you know that it has the operator region, the promoter region, then it has the inducer region, the LAC-Z gene, the LAC-A gene, the LAC-I gene and all that. So I have to write what is the function of each of these genes. So I gene codes for the repressor of the lac-operon. LAC-Z gene, it codes for the beta-galactosidase. Then LAC-Y gene, it codes for the enzyme permease. LAC-A gene, it codes for trans-acetylase, which actually, and here inducer is nothing but the substrate, that is lactose, for the enzyme beta-galactosidase, and this inducer is the one which actually switches on and switches off of the gene. In the presence of an inducer, the gene will get switched on. In the absence of an inducer, the gene will get switched off. So much you have to write about this. Next question, briefly explain the uses of transgenic animals. So here 5 uses need to be written as it is given in the NCERT textbook. So the first use is study of normal physiology and development. So transgenic animals can be designed in such a way that it can be used to do genetic study, that is gene regulation, and what are the functions of the gene and all that can be studied. Next is study of diseases. So specifically designed transgenic animals can be used to study diseases. So like we can understand about those diseases such as cancer, cystic fibrosis, rheumatoid arthritis, Alzheimer's, and the whatever output we get or the results we get from the experiment, we can incorporate that to treat the patient. So for that it is useful. Next is production of biological products. So transgenic animals are designed to produce biological products such as alpha-1 antitrypsin, and also we can get milk enriched with nutrients like alpha-lactalbumin that are used in medicines required for the treatment of various diseases. So therefore, to make biological products to treat diseases also, these transgenic animals can be utilized. Next is for testing of vaccine safety. So transgenic mice can be used in order to test the safety of a particular vaccine for a particular disease before it is given to the human. So for that also it can be used. So say for example here they have tested the safety of vaccines like polio vaccine before they were administered to the humans by developing transgenic animals. So 4 points are done. We need to add one more because it is a 5 marks question. So the next is testing of chemical safety or toxicity testing is what we call it. So transgenic animals can be made which carry genes that are designed to be more sensitive to toxic substances so that experiments can be done on those animals to test the safety of that particular toxic chemical. So for that also it can be used. So next question, what is mutualism? Explain any 4 examples of mutualism. So we need to write the definition of what is mutualism. You will get 1 mark, and if you write 4 examples with explanation, you will get 4 marks. So a total of 5 marks. So mutualism is nothing but the interaction that confers benefits on both the interacting species. So both the species which are interacting with one another, they derive equal benefits. So that is called as mutualism. So one of the best example that we can take of is lichens. Lichens have a close symbiotic or a mutualistic relationship between the fungus and the photosynthesizing algae, that is the phycobiont and the mycobiont component of the lichen. Next is mycorrhiza. Mycorrhiza have a close association with the roots of higher plants such as pines and all that. And these mycorrhiza, they help in absorbing phosphorus or other mineral nutrients in a faster rate from the soil. So that is one of the example wherein the mycorrhiza is also benefited because the roots provide a home for the mycorrhiza, and the plant is also benefited because it can derive more amount of phosphorus from the soil. Next example, fig species can be pollinated only by its partner wasp species and no other species. So whenever we cut open a fig fruit, we can see larvae and flies inside it. That is because the pollination happens inside that itself. So therefore, the female wasp uses the fig fruit for laying the eggs, and during that process, pollination will also happen. So therefore, it is a mutualistic relationship where the fig can get pollinated, and for the wasp, it provides a safety place for the laying of eggs, and also once the eggs hatch and the larvae can come out, they can use it as a food. So therefore, it is a mutualistic relationship. Next, fourth one, the Mediterranean orchid Ophrys employs sexual deceit. So sexual deceit is nothing but wherein the flowers, it resembles or it starts looking like a female insect, so that the male insect can come and sit on it for mating purpose, and therefore pollination will happen. So therefore, or sexual deceit is also one of the examples for mutualism. So this is how the answer needs to be written for that. Moving further, we'll talk about the next part that under part D, we'll talk about the next session, that is main 6, wherein we need to answer any one of the question out of the 2 questions that are given. So the first question, identify the genetic disorders given below. So they have given 5 genetic disorders. We need to identify. So here they have given 5 statements regarding the genetic disorders. Reading the statement, we need to find out or answer what type of genetic disorder it is. So the first one, sex-linked recessive disorder occurring in about 8% of males and only about 0.4% of males, which leads to defect in the cone of the eye. So defect in the cone of the eye, it is nothing but color blindness. So the answer for this is color blindness. The next one, sex-linked recessive disorder affecting a single protein involved in the clotting of blood. So when you see here clotting of blood, when you see here defect in the cornea of the eye itself, you'll understand. So one is color blindness. When you see it is clotting of blood, of course it is hemophilia. So the answer will be hemophilia. Next question, inborn error of metabolism, autosomal recessive trait, accumulation of phenylalanine. So when you read this line, that is accumulation of phenylalanine itself, you'll understand that the answer is phenylketonuria. Next one, autosome-linked recessive blood disease, substitution of amino acid. That is from glutamic acid to valine. So that is nothing but sickle cell anemia. So therefore, the answer is sickle cell anemia. Next is autosome-linked recessive blood disease, reduced rate of synthesis of globin chains of hemoglobin. So that is nothing but alpha globin chain and beta globin chain. There are 2 types, right? Alpha thalassemia and beta thalassemia. Here they have not mentioned about the chain in particular. So we can write thalassemia as the answer. So this is about the answer for this particular question. Next question. The flow chart shows replication of the retrovirus in the host. So when I say retrovirus, here we are talking about the human immunovirus, that is the HIV, right? So here the answer, fill in the missing data in the boxes labeled 1 and 2. So here first one is viral DNA is produced by reverse transcriptase. Second one is new viral RNA is produced by the infected cells. So give an example, that is the first question. So next is give an example for retrovirus. Here the example for retrovirus is nothing but human immunodeficiency virus. Next is name the disease caused by this retrovirus. So the disease caused by this retrovirus is acquired immunodeficiency syndrome, which we commonly call it as AIDS. Next one, name the diagnostic test for this disease. So it is ELISA, that is enzyme linked immunosorbent assay. It is ELISA. So this is the actual table that is given. So here 1 and 2 has been left blank. So first what happens, retrovirus, it infects the normal cell. Viral RNA is introduced into the host cell, then reverse transcription happens. After reverse transcription happens, the viral DNA gets produced. That is the process of reverse transcription, right? From the RNA, DNA strand gets produced. So, the viral DNA will get produced. Then viral genetic material is introduced into the nucleus of the host cell. Viral DNA incorporates along with the host DNA or the genome. Next, what happens? Next, new viral RNA is produced because again their transcription mechanism will happen. So therefore, viral RNA will get produced. Viral RNA produced. Viruses come out of the host cell and then the new viruses will infect the other cells. So that is usually what happens. So that is what we wrote the answers here. So we answered A, B, C and D. So this will carry 1 mark, 1 mark, then again 1 mark, 1 mark, a total of 5 marks. Next, give reasons for the following. Restriction enzymes are called molecular seizures. Why are they called molecular seizures? Because they cut the DNA at specific locations. That is why restriction enzymes are called as molecular seizures. Next, under this question, the sub-question, DNA ligase is used in genetic engineering. Why is DNA ligase used in genetic engineering? Once the DNA strands are cut, the lagging strand, we need to join it. Right? So, to join the strands, we need the enzyme which is called as ligase. So, therefore, for that also it is needed. And not just that, in order for linking of antibiotic resistance gene with the plasmid vector. So, for that also ligase. So, any DNA strand we need to link anywhere, we need ligase as the enzyme. Next one, chilled ethanol is used in DNA isolation because purified DNA ultimately at the end it precipitates out after the addition of the chilled ethanol. So, that is why chilled ethanol is used at as the last for spooling, that is process, chilled ethanol is used. Next, smaller DNA fragments move farther in a gel slab. Smaller the DNA, lighter it is. So, therefore, lighter materials tend to move faster when compared to the heavier ones. So, that is why the gel electrophoresis, the agarose gel in the gel electrophoresis, it acts like a sieve and therefore, the smaller fragments can navigate or move further through the pores in a faster rate when compared to the larger ones. Next is thermostable DNA polymerase is used in polymerase chain reaction because it remains active during the high temperature and it will not get denatured. So, that is why thermostable DNA polymerase enzyme is used. What is the function of DNA polymerase enzyme? In order to add the nucleotides to the newly synthesizing DNA strand, and it should be heat resistant or thermally stable. So, that is why thermostable DNA polymerase is used in PCR. So, this was about this session wherein we learned part D, that is section 1 and section 2 under part D consisting of 5 marks. I hope you found this session quite useful. So, with this, the model question paper discussion comes to an end with a total of 3 sessions. Thank you.